So for today, I thought I would spend maybe 45 minutes talking so we have more time for discussion. But I wanted to go over the whole program overview, some of the importance of phytoplankton in the Antarctic Peninsula. So how many are biologists? Okay, good, because I know Polar Seminar typically tends to not have biologists highly present. So I have a whole section to describe more about phytoplankton that hopefully won't be too dry. I'll talk about the framework and the methods for how we analyzed the phytoplankton community in Antarctica and some of the highlights of the emerging seasonal and interannual patterns between the different years. And then I want to end on spending some time showing you guys this virtual field trip and also kind of show you the back end. So if you wanted to use this tool to make a virtual field trip for anything you're working on, there's a lot of cool custom ways you can do something like that. And I'll just show you the way I've done it to kind of mimic an experience in Antarctica. So FjordPhyto is a participatory. You may have heard citizen science. I think in general, the community is moving away from the term citizen for mostly US political reasons. So participatory seems to be a common way to describe it. But it's developed in a partnership between the tourism industry in Antarctica that is governed by this organization called, the acronym is IATO, but it's the International Association of Antarctica Tour Operators. And they're bringing travelers to the polar region, to Antarctica. And FjordPhyto's aim is to study the phytoplankton community changes due to a changing environment, while also having a positive impact with the tourism industry. So we feel together we're creating this knowledge, we're making discoveries, and we're providing life-changing experiences for many of the people who do participate. Of course, they're also in Antarctica, which is life-changing in itself, but we like to think participating in a little bit of science is helping to that. So participatory science is the practice of engaging the public in a scientific project that produces reliable data. Hello, for those of you who just joined, and information usable by scientists that is open to the same system of peer review that applies to conventional science. SciStarter.org is an amazing website that lists thousands of citizen science projects or participatory science projects. So you can search by region or topic or program if you want to navigate through that and just see sort of the breadth of projects that exist. And of course, we all know in Antarctica, there's many people going down there, 33 national Antarctic programs. And if you want to gain access to the field, it's pretty difficult, especially now. This was a slide from my defense in December last year. So now we say goodbye to the research vessels. And on the peninsula, I've just zoomed into the section that shows where the different year-round stations are in yellow, as well as the summer-only stations in red. And then the blue stars the US Palmer station. So you can kind of see the spread of where research-based stations are and whether they're doing research in the summer months or year-round. And there's also, of course, the travel industry. So IATO is established in 1991, and it contains about 100 private sector operators at the moment from around the world. So your ship can be flagged from any country. And they come down to Antarctica November through March and every single year. So they're heading down south now. Actually, some are already visiting South Georgia. And it's an untapped potential with these ships of opportunity that you could work together with them. And it also is like an immediate community of people to engage with in sampling efforts, but also education. So this contains the expedition vessels. It contains the expedition guides that are hired on board the ships to be working the whole season. And it contains, of course, the travelers who come down on vacation. Has anybody gone to Antarctica on a expedition ship versus a research ship? Christian? Yes, Christian is very familiar with that. So the expedition cruise sector provides this public engagement because they're bringing people every 5 to 15 days. And it's reaching in 2022 to 2023, the visitations were 123,000 individuals that actually traveled down. When you think of like national parks, you know, some of the national parks here get 4 million visitors a year. So in context, maybe there's some discussion on is travel there saturated? Should it spread out more? What kind of regulations should happen to kind of sustain the growth of people visiting? So on the left here, this image is showing from 1992 all the way to 2022 to 2023. When I started at Scripps and we designed this program, there were only 47,000 people visiting. The pandemic, of course, nobody went down. And then it's just skyrocketed since. So they built 21 new vessels after the pandemic. And I meant to change this because the US now has zero. So we were going from 2, then we went to 1, and now we have 0. So if you compare the number of visitors, you also include all the people that are working on the expedition ships, in contrast to people who can go down through the National Antarctic programs, either through the US or Argentina or Chile, or like whichever country runs a program. Estimated COMNAP told me that was roughly 10,000, could be 20,000. It's just very difficult to determine because it's the length of stay isn't integrated into that. So it's like estimated 10,000 to 20,000 in the summertime of science support people going to the national research bases or on research vessels. So over the 9 years, this is a mosaic of all the different expedition ships we've been able to work with. We've partnered with ships like you see down in the bottom row. Bark Europa is a tall ship, wooden. So if you want to sail back in the olden days, you can hitch a ride with them, all the way to sailing yachts of 12 passengers that come along, all the way up to some of the bigger ships that hold 500 passengers. So a lot of this is determined based on the Antarctic Treaty, and the regulations state you can put 100 people on land at a time. If your ship has more than 500 passengers, you're not allowed to make a landing at all in Antarctica. You just have to cruise by. So a lot of these ships are within the size class range of about 200 passengers, but there are some that are bigger with 300 or 500 passengers. And so this is sort of the example of the ships that are going down. So FjordPhyto itself, what does that entail? So we provide the expedition guides training specifically for this program, and we provide all the needed gear and the materials that they can use for this. The travelers are called guests, guest engagement. And it's a curated 1-hour science boat experience, because when we were designing this, we couldn't, like typically as a scientist, you want triplicates, right? You want all the data possible, but in the collaboration, they don't have that amount of time at a landing site to be doing triplicates. So 1 hour was the kind of the sweet spot for what we could design as a program. They could collect data that helps us answer questions, and that was sufficient. And this is contributing to real science. So there's a lot of this negative attitude towards participatory science as it's not real. And I like to think I'm doing real science and that they're doing real science. And so that is where the definition comes in, that it's held to the same standard of rigor. And there are different ways you can kind of have checkpoints for that. This also, of course, has the educational content, the workshops that are provided on board, as well as with our program, we provide real-time responsive support and open communication. So we try to be very active on the WhatsApp channels with each ship and each guide during the season. Back in the day when internet wasn't as good down there, now internet with Starlink seems to be better and better. You can actually FaceTime people from Antarctica. You can't really go off grid as much as you could in even just 9 years ago. So, but this is good because if things happen and you need to troubleshoot immediately, you can. And we try to adapt the activities for every operator. So we have our standard protocols, but it can adjust to how they run their trips. So I published the methodology and kind of the reasons behind this and how I developed this program. This was actually a part of my master's degree when I was here in 2016 for the Marine Biodiversity and Conservation Program, MAS program. Which I think is amazing. And if you want to collaborate with any MAS students, are any MAS students here? Like, it's an amazing cohort of students that are very interdisciplinary. So tap into that program. But this publication talks about the toolkit we walked through to really think through all this. So our online training is one of the ways we ensure that we've rigorously trained the individuals, the guides who are facilitating this program with the travelers as they come and go. So we have, you see one of our stars here, Christian is in the audience, not to point you out. So we offer a pre-assessment to kind of gauge where they're coming from and what their background level is. And then they go through our online training, which we've hosted with NASA's Infiniscope program, which is what I'm going to talk about later. They do digital learning, so we do our training with them, but the field trip I'll talk to you about is also through the program Infiniscope. So this is sort of intended to be like a choose your own adventure style. So it's not just like, well, hopefully we don't think it's a boring training lesson online, that you can have some clickability and learn as you're going through learning how to do the science. And then the post assessment tells us we can then see if we're able to like gauge if they enjoyed it, but also if it's having the effect we hope it's having after they do the online portion, and they get a certificate. Because we're also noticing in the industry, a lot of the expedition guides get hired now as citizen science coordinators. And if they can show they know how to do these types of programs, they put that on their CV, which when I first saw that, I was like, oh my God, that's so cool. So we're like, okay, we can offer a certificate that ensures you have something that shows you took the online portion. And then we follow that up every fall with in-person training. So we go to port in Argentina where most of the ships are coming in and out of, and we have physical people handing the gear and doing physical training with the ships as they're coming to port. And then if people are on board the ship, like when Christian goes down or I go down or my colleagues in Argentina, they can also continue to do the in-person training on the ships. So that's one of our ways of having kind of a safety net to ensuring rigor. And we have super guides now too that we trust to train others as well. So what are people actually doing? So they will take one of these rubber inflatable boats. We just call them a Zodiac. It's kind of like we say Kleenex or Q-tip, or it's just the brand, but it's a rubber inflatable boat with a tiller. Some of them have center console. That doesn't matter. The point is about 6 to 10 people can fit. Some of them get up to 15 people at a time, if you have a little bigger one. So they'll go to a certain location. Typically, we've matched them all up to where the main ship is going anyway, so they don't have to go out of their way. And if the GPS location we give them is more than half a nautical mile, like 1 nautical mile is about 15 minutes of driving, we do not go there, right? It's just, the idea is it's convenient for operation. So they'll go to these locations, but that ship goes repeatedly every week, every 2 weeks. So we kind of build this time series. And in that boat, they're collecting information about the environment. So they're dropping a CTD, conductivity temperature depth device, down to 100 meters. We get the physical oceanography data. So we have the salinity, we have the temperature, chlorophyll A on some of the sensors, have a Turner fluorometer. And then they send over a Secchi disc. We just use the black and white Secchi discs because we're in more of a coastal region as opposed to the open ocean. And that gives us light level, turbidity information. Sorry, I should be using the pointer. CTD, Secchi disc. Then they physically collect samples from the water. So they'll do bottle samples where we can send these off to our colleagues in Argentina, Universidad Nacional de La Plata. And that team will analyze the sample under microscopes. So they get cell abundance. They can identify the species because they're traditional taxonomists, and estimate the amount of carbon biomass, which is important metric, of course, for carbon cycle and food energy available to the system. And then they tow a phytoplankton net behind the boat and filter that down onto a filter. And those filters come back to us in San Diego up at the James Craig Venter Institute in Andy Allen's lab. And that's where we're processing all that data for DNA and RNA. So at a genetic level, how are they responding? And then we can do species identification also with the DNA. And then the idea is, for the last 4 years, we had funding from NASA. So we were ground truthing what satellites and remote sensing sees because we're physically in the water collecting the samples. So over time, this is, very tiny, but we've got the different seasons. I haven't put in the last 2 years. So we have 6, 7, 8, and this season marks 9. And the frequency of the number of samples collected at each site gets warmer into the green and yellow colors. And I've just flattened all the locations. So if you were to go from the south end of the peninsula all the way to the north, you would kind of like follow the spine of the Antarctic Peninsula. Originally, they were only going in the central region, but then as we got more ships joining, we started off with 2, then we had 5, then we had 15. It just kept growing. So they expanded where they were exploring and also where we were taking samples from. But you can kind of see that sort of, in some places, increase the amount of effort for sampling, but overall kind of reduce the sampling intensity because they're not just visiting the same sites, they're expanding that. So over this season including, we've multiple sites are being sampled. That's given us thousands of samples. We don't have enough people to analyze all of this rapidly. And we're creating this large spatial scale sampling as well as that time series, November through March, every single year. And we've presented this, all the results that we've had at over now 30 scientific conferences. So we're really trying to like go see people in person, go to the different communities, scientific events, participatory science conferences, as well as social science conferences. And we've published in 9 peer-reviewed publications. So if we talk about publication as the currency of science, like we have that peer review process, putting out the results of the data. And one of the fun things that is just come out this year, I'll have a slide on that, is a new organism that was discovered. So I'll talk a little bit more about that. So that's why we really do feel we are creating knowledge and together with this program. So it's all people powered, right? And we've got 16 operators, 26 vessels. We've trained over 100 expedition guides. They've engaged over 10,000 travelers. We just estimate if you're fitting 10 people in the actual boat, how many. This has allowed data that is not only my PhD, but we had my colleague Martina in Argentina finished her PhD, and we have 2 new PhD students continuing, one in Argentina and one in Chile. And then this has also provided opportunities for undergraduate students and for summer surf program or internship opportunities, either to process samples in the lab or to analyze the data on computers, and as well as the master's students, the MAS program. We've had over 10 MAS students working on this program, which is really why we have a logo. It's why we have a phytoplankton identification guide, a teaser video, an 8-minute long video. It's why we have educational material. Like everybody's brought in their own element of the collaboration and really expanded it. And then, of course, we've worked with social scientists now. Artists have come on to work with us, filmmakers, educators, and more. So this is really getting a life of its own. It's very weird. It feels like a child that's like running and you just have to like figure out how to keep feeding it because it's growing. So one of the participant findings is that travelers are highly valuing this experience. We offer a survey. Initially, the first 81 we ever collected from the first year or 2 of this program, we ended up publishing in the Polar Journal about the results of the experience being enhanced for people. They feel like it allows them to be more than just a tourist. There's a lot of guilt in traveling community, especially on cruise ships. And so a lot of people feel it also reinforced Antarctica as a crucial part of climate change. And that integrates with like the lessons they're learning while they're down in Antarctica anyway. But this is an element that adds to that. So a master's student designed an official survey for us, taking the IRB exemption and the classes. And we had another master's student then analyze 300 of those survey results. And she just presented that this June for her capstone. So we were working on publishing the results she had, but it was a YouTube video you can see, learning on vacation. So we have a YouTube channel. We try to post as much as we can there. So we're transparent what's happening, as it's a very public funded, public facing and public collected collaboration. So that's very exciting. That has been one of the things that comes out of it. But what was interesting looking at those first surveys was that 97% of the respondents said that enriched their travel experience. But interestingly, 62% said prior to booking, they didn't know that these projects would be available to participate in, which I find interesting because a lot of the operators we work with do advertise that they run these types of programs. So I'm not sure what some people book through booking agents, like, you know, there's a gap there. But 65% of the respondents said that the availability of those projects on their trip would influence their decision to book a trip over another. So there is a desire for people to have these kind of more immersive educational experiences. Also, the expedition guides, as I mentioned, they're putting it on their CVs. We've trained over 100 of them. We really tried to tailor it so that anybody could become a guide. And last season, 60% of the people who took the training were new guides. They didn't have, I put master's degree or higher, but we had bachelor's as well. The idea was you don't have to have a science background to learn how to do this project. And then, of course, you can engage in the environment, but also lectures on the ships. The media is loving it, so that's just good for more exposure because then it helps us find funding and people talk about it and know about it. And speaking of funding, I like to point this out because I know, especially in the stage we're in now, everybody's kind of struggling with funding in the situation. So in the last 9 years, if you total up the funding we had to start it from the National Science Foundation, as well as some fellowships we've received from the Antarctic community, the tourism community, donations, auctions, foundations, NASA has been very supportive with a longer term grant we had. A lot of this outreach grants come from a seed grant program they have, which I'll talk about because it's great for early career scientists. And then a Chancellor's Innovation Award recognized the program. So all of this has happened with under $2 million, which I think is incredible because you start putting price tags on science, and I think we could use 2 million a year. Everybody says that though. Then think of what we could do. And then, of course, a lot of the people working on this are student stipends, right? It's nobody's full-time paid for this. This, thanks to Garrett in the back, made a quick, kind of like trying to put, actually UNESCO asked if we had an infographic, and so we didn't. And here's another example of a collaboration forming, is like, we can work together to create something that might now start helping integrate what's this program and the data that's come from it into management and policy. Like that's sort of the next step that it could take. So it's just showing data flow, like the metadata, the environmental samples go into Dryad. We're currently publishing that and making it findable, accessible, interoperable, reusable. The DNA data we're storing in NCBI. So the first 4 years are up there. And I'm currently in the lab processing samples with one of the undergraduate students for the last 4 years. Microscopy data is all going into the Global Biodiversity Information Facility, or GBIF, as well as some photographs in iNaturalist. And then the idea is, have you guys heard of SEUS, the Southern Ocean Observing System? So they have the SEUS map. And ideally, this could be a contribution to the SEUS map efforts. Also working on a cool initiative and idea for a database that highlights integrating all this data globally with the Walter Munk Foundation. So that's an exciting also development in the works. So that kind of wrapped up the first part. The next part, I'm going to focus more on the results of the science that have come from and the phytoplankton and what we know about the seasonal patterns emerging. And this is going to be a little quicker. I kind of felt like I might want to set up the whole program so you can see the framework of where this data is coming from. So the main objectives of the research were to understand how phytoplankton communities are being affected by climate change effects, so melting glaciers, warming temperatures, and understand how the phytoplankton community is changing through the spatial scale and time, and also how they're composed, like which species are making up the community. And why do we study phytoplankton? Of course, I mean, you know, even though there's no biologists, I know we all know that phytoplankton play a critical role in drawing carbon dioxide out of the atmosphere. They're the base of the food web. They're converting the organic food for like krill and copepods and amphipods, and they contribute to producing over half of Earth's oxygen, which fluctuates. So we've got a schematic here showing, of course, phytoplankton, and we're going to go right to left. So the photosynthesis happens, and this is the Antarctic simple food web, feeding the krill, supporting it under the sea ice, but also in the open ocean. That then falls to the benthos, supporting the benthic community of organisms living on the seafloor. And then this krill, the keystone species or key species in Antarctica, is then fed upon by whales, seals, squid, penguins, seabirds, and fisheries. So it's really incredible, this strength of this ecosystem. And phytoplankton do come in different shapes and sizes. So here's a smattering of different sizes. On the tour ships, of course, we just identify them as pizza-shaped, football-shaped, hamburger-shaped. Usually we're always hungry because we've just finished So cactus. But this allows us to really talk through. It's like you have birders. Birders are insane with their bird knowledge. You have people who love plants, and so we thought, why not have that same enthusiasm for phytoplankton? They're knowable. You can get to know them. And we tried to demonstrate the size difference. So we always talk about phytoplankton like they're all kind of generic, but they range in size. If you were to think of a zooplankton, a copepod, which feeds on phytoplankton, we have a different types of phytoplankton listed. So some diatoms, Cocinodiscus, Porosira, Catoceras, chain-forming diatoms, as well as a non-diatom, a Phaeocystus. And if you were to relate that size to something we might be more familiar with, you're comparing an anchovy to a blue whale. So the size difference between phytoplankton themselves can be up to 100 times. So if you really think about that as food storage, you have krill wanting to feed on massive diatoms that are big and full of lipids and omega-3s and really rich. But if your whole food web is changing to smaller phytoplankton, you're eating anchovies. Of course, we know blue whale can exist on krill, but right, like you have to think of the scales, even within phytoplankton at the microscopic level. And we also tried to convey this with this infographic too, is like if you were to compare a big tree to a tiny weed, that might be similar to comparing the scale of a big diatom to a tiny cryptophyte flagellate. So if you're a krill and you're trying to feed upon, you have your feeding strategy to scoop up, oh my gosh, if you have not seen krill teeth, you should Google krill teeth. Why have I gone my whole time here and not seen a picture of that? They're crazy, like jaws with silica lined on the tips of the teeth, and then they just crush the diatoms and break them up. So if you're looking for that as your food source and all you find are these tiny little things, like that's going to be very disappointing. So that will also change the global carbon pump. And I know we talk a lot about chlorophyll, right? We talk about high chlorophyll, low chlorophyll. Here's on the bottom is an example of high chlorophyll. Sometimes in Antarctica, it's like soup. It's so thick. And then this would be a lower chlorophyll, less phytoplankton. But what we want to know is the actual community, exactly because of what I just said with the size and the actual species present. And the idea is that we can identify succession. So you have phytoplankton blooming, you know, similar out here. You have the community blooming on the coast, but then also how is that changing seasonally? The typical idea is that it starts with sea ice diatoms, those melt away. You have your diatoms blooming, then you get a mixed community, and then it transitions to these smaller cryptophyte, those little teardrop shaped ones. That's sort of like the hypothesis in the literature right now. And that can happen over one growth season. But when I really dug into the story of what is happening, is this transition seen all over the peninsula? Some of the main observations that are reported is that the community is shifting away from the large phytoplankton to the small. The community diversity within who's making up that is declining. Previously unobserved bloom-forming species are starting to dominate. So there's more occurrences of certain phytoplankton blooming in massive numbers than previously seen. And previously underreported groups of phytoplankton are also becoming dominant. Before seen, like even we've discovered something that had never before been seen in over 50 years. And studies are showing the community structure and the shifts are driven by, of course, the environment. So that can be very complex though. It's like, is it the sea ice? Is it the light, the nutrients? Of course, we're familiar with this story. That's what we do for a living, scientists analyzing this data, trying to figure out what is the driver. And the divergent results in the literature, one study can publish one thing and another study publishes a complete opposite information. And why might that be? We think it's just because of the uncertainty due to spatial and temporal undersampling. So if you think back to where the research stations are located and some of the long-term LTER, for instance, is 30 plus years, but it's on the continental shelf. It's off the coast, and all of the research stations are on the coast. So they're taking measurements at research stations. And so you have some that are able to do those time points. But in general, if you look at the sampling effort for Antarctica, we know it's the data limited region in the world. So one of our questions that led us to this is, with partnering with the tour vessels, can we expand that coverage and sampling efforts? So that brings me to, that was the importance. Now I'm going to just dive into some quick highlights of the microscopy, the DNA, and what the seasonal interannual patterns showed. So again, this is our sampling strategy. They go out in the boat, collect all this data. One of the samples collected from the seawater is the brown bottle stained with a Lugol iodine dye that's sent to our colleagues in Argentina, and they look at this on the microscopes. So we have a past PhD student, future or current PhD student analyzing 4 hours for each sample, I heard. So it's very manual effort. And then you have to get good at identifying what the heck is this? Like who is who? How, you know, it's a whole specialty. So that is dedication. And that's where they get the information of cell counts, species identification, and carbon estimates. Then in the lab, those samples that were collected with the net, that jar is filtered. So this patty, this cake of phytoplankton is rolled up in a tube, and that tube has a RNA later preservation solution just that helps it not degrade. And those get sent back up into the lab. And again, that's a lot of manual labor to take the tube, take out all, like this one is almost black. It's got so much phytoplankton in it. So you want a portion of that. You can then extract the DNA or the RNA. You can do PCR amplification. So I'm currently working on 2,000 PCR amplifications. And you just target a specific part of the gene that is well conserved. So when you do like taxonomy, you can tell if it becomes a different species, organism, like where it fits in the tree of life. And so using certain primers or markers or barcodes, there's like so much jargon in the genetics world. You can look at either prokaryotes, bacteria, plastids of phytoplankton, or phytoplankton. But you also get back data on eukaryotes. So we do have data that comes from any 18S organism. The data set also has crustaceans and mollusks and fungi. So we just take out the part on phytoplankton because that's what our research questions were. And so those samples get sent off to the sequencing centers up at Upper Campus. There's 2 different sequencing centers we use, the G-Core and IGM. And then they send us back probably within a month. We get back all of this data, FASTA files. It then just turns into computer life. So we do bioinformatics, use different programs, the Protis database and a bacterial database called Silva that helps identify which gene, which DNA is which organism, how well does it match, and then we do our data analysis and interpretation. So that's a whole fun thing that AI has really helped in coding faster. One of the highlights, so this is showing that timing really matters. So in this figure, I have the first 4 years of DNA data, one of those primer sets. So I'm looking at the eukaryotes and clustered them based on what kind of phytoplankton kind of dominated the results. And so I've got the small phytoplankton cryptophyte, mixed diatoms, and then the bloom forming ones, or these like rare ones that are bigger. Each point on this is a science boat, like an activity where humans were in the boat collecting the sample. And you can kind of see the scatter over the time. So I've just turned time into 0 to, what did I go up to? 150 days. And that kind of covers the season. And then the first 4 years show the proportion. And you can tell if you were to go the first year and sample on day 50, you would see a high mixed diatom community with the bloom forming diatoms next. And then the cryptophyte community, or those small phytoplankton, are very small. If you then went and sampled a month later, you would get a different collection of phytoplankton in that community. Now, if you look over the full 4 years and just look at the gray line, it does seem the trend is lower small phytoplankton in the beginning of the season, and then they increase towards the end of the season. That's what we think, that's what we know happens. But you can see it varies depending on which year. Sometimes they crashed, sometimes they went up really high. But what's interesting is if you're sampling and you see the mixed diatom community, let's look at the second year, you would go sample, you would think your community was only these big bloom forming diatoms 50 days in. So of course we know seasonality changes, who's going to be there when. But you look over just the 4 years, the data is more sparse, of course, because of the pandemic. All the ships went home and didn't come back right away. So now it'll be interesting when we complete the next 4 years, we'll have this big 8 year, we'll probably wait till we get this season. So we'll have 9 years to look at what is this trend? Is this a trend? What's happening? Because we all know too, a lot of the science in the reports are the length of somebody's PhD project or the length of funding from 3 to 5 years. And so there's this like effect where you only really get a chance to look at a couple years in coastal information, unless you're, I know Rothera, like the British, has an amazing year-round sampling. Carlini Station, the Argentine station, and Palmer Station as well from the US. Either way, this is explained in more detail also on our YouTube channel. I haven't yet published. I'm working on submitting it. So one chapter is ready to be submitted, and the next I'm trimming from 10,000 words in the dissertation to 5,000 words for submission. So again, this also can help decipher, like we're seeing chlorophyll from satellite images. I'm super excited about the PACE mission as well. Like PACE mission can tell you the different groups of phytoplankton, so we can then relate that to the groups we're seeing. Another fun result was, are you guys familiar with the Gerlache Strait in Antarctica on the peninsula? There is a region where there's a front. It's a thermal front, so the temperature is meeting. So you have the water coming from the Weddell Sea around the north end of the peninsula, and then you have the circumpolar current that goes around Antarctica is slightly warmer. It comes up on the shelf, and they meet right here in the Gerlache Strait. And this, of course, causes upwelling. This is where you see a ton of humpback whales feeding, especially at sunset. It's a hot zone for different sites to bring the ships. And so in red, that's indicating a warmer water, and the blue is indicating cooler. So comparing just communities in those different locations, my colleague Martina also saw that just looking from the north where it's warmer to the south where it was cooler, kind of compiling all those results, the baseline of phytoplankton that exists is this basic assemblage. But then again, you're seeing different phytoplankton kind of blooming, and this purple one sets off the both locations. But then what follows is completely different based in the warmer water or the colder water. In the colder water, you have more of these large diatoms. In the warmer water, you do have more of these smaller phytoplankton. So that's what we're able to really look at with this time series. And then the new species, so this was fun. It had never been described, a naked dinoflagellate. So this is what it looks like under the microscope. I think, you know, when you're a taxonomist and you're staring at the microscopic level for hours and hours and hours, and you see something that isn't what you're used to seeing, this is very exciting. As a person analyzing genetics samples, I don't get to see any of them like this. I just see brown. So and like data on a computer. So this is very exciting too. And she shares photos under the microscope. But combining, right, you can't also just write a paper now using morphology under microscope. You have to pair it with genetics. And so yeah, that's how we worked together on this, was a fellowship. She got to come to the lab and kind of pair this effort. So yeah, there was just a ton of them growing. It was one of these blooms that hadn't been documented, and the genus hadn't been studied in Antarctica for more than 50 years. So never been described. I think there was some mention of it in maybe somebody's diary. I don't know. They hadn't like really picked up on it. So that's exciting. And that just came out in press. So that brings me now to what I think is the more fun part, the outreach. So right, we've engaged this community in Antarctica, which has been a huge success and been very exciting and very like remarkable to be able to build this collaboration. But then we think, what if you can't ever go to Antarctica? What if you don't have the opportunity? What if you don't have the means? It's definitely, you know, higher income-based individuals who are obviously able to afford a trip to Antarctica, which can run anywhere from $12,000 to $50,000 or more. So, you know, for a 5 or 15-day voyage. So it was sort of like our effort to bring what we're doing down there to more people who can't go down there. And one thing we really see is that like Antarctica is so remote and distant from many people's minds. It's really hard to relate. Like, why would you care about Antarctica? Particularly, we teach in Puerto Rico. They're learning about their tropical ecosystem like they've never seen ice or glaciers or snow. So how do we really connect the poles to this more global view of, because we know it's the hub, it's the air conditioning system, it's, I think the heartbeat of the ocean. So one really cool opportunity that I hope you guys all check out, and I hope it survives whatever's happening with this shutdown. NASA science activation teams. Have you guys heard of these? So NASA has mandates to have teams that are solely to educate and expand NASA missions through outreach. And so there's like a huge list of programs. I just put a couple here to show you. But the idea is it's NASA science experts, content, and experiences connected with diverse learners of all ages to foster a deeper understanding of the science and increase people's active participation in the advancement of human knowledge. And so Arizona State University and NASA have partnered to create this seed grant opportunity called NASA Scope. And the idea is that if you are somebody who, an early career scientist who works with NASA data or for a NASA-funded program, you are a subject matter expert, a SME, and you can partner with one of these science activation teams to then write a two-page grant proposal, submit it through this NASA Scope portal, and they also offer grant preparation. So if you want to get kind of more guided training on how to write a grant, it's just a two-page one. It's not like one of the 15-page ones you send to NSF or NASA or anything, but it's very intended to like support the early career scientist journey, and you get to be the PI. So it's also I've been fortunate now to receive 5 of these, so it just kind of boosts you a little when you're like, I'm a PI, like I'm not a little grad student, like so. I love this program, and you get to meet so many cool other outreach programs. So have a look through their list. We've partnered with Oceanos, which is how we go to Puerto Rico the last 3 summers to teach there. And Infiniscope is the platform we partnered with to create our training, but we've also now partnered with it. Infiniscope's idea is to bring digital teaching, build the network. So NASA-funded project at Arizona State University with the mission to transform Earth and space science education. And it achieves this by providing educators with free digital tools, so you can go online and sign up freely. You don't need a grant from Scope to start this if you want, or if you're a teacher or anybody. And there's professional development to create engaging adaptive learning experience for students and the NASA data expertise. So one of the tools they have is creating a virtual field trip with a program they've called Tour It. And so they have a lot of like learning lessons, but the idea is that if you could create a virtual experience, does that give people the same sense of learning and care for a place? And they've done some studies and looked at different field trips they have, and there is this power of place. People do have place-based learning. So like if you're in a place and you learn something about it, they call that place-based learning. You've learned a lot. So that's what we're doing in Antarctica. They're learning about all the things surrounding Antarctica and maybe having stronger memories and changing behavior when they go back home, like whatever they feel inspired to do. Can you get that from a virtual field trip? And the answer, what Infiniscope's found, is yes, it can have the same effect, probably similar to VR, right? Virtual reality worlds and feeling real. So I wanted to show you what I've created. We're at 2:46. So I think I have to exit out of this. Well, I'll show you the back end first, and then I'll exit out. And so basically, if you have any 360 photo, sorry, or a panorama, you could do with your phone where you just take a panorama, or if you have an Insta360, I bought an Insta360, you can take a video and then like export just a flat image. You upload all those images you want, and then you go in and you then annotate them all. So the idea is you're going to enter this experience and kind of be guided through it, but you're going to move from scene to scene. So they have a lot of examples online, but of course I'm going to show you this one. Are we still saying, okay, now I gotta move all that. Can you hear the sound? No, how do I do sound? Okay, so, well, either way, this is the page you land on. There's penguins chattering in the background. I don't know how to do the. Okay. And the audio. More. Audio. Uh-huh. Oh my gosh, really? Wow. Ugh. Well, okay. Sad. Just imagine penguins chattering. Wait, how do I? Sorry. There we go. You hear that? What is my password? Okay, well, here to my computer. Right, got some chin strap penguins pattern. You can get rid of this thing and then swirl around. You can hear footsteps. Oh gosh, I'm in the sky. You can place these hotspots anywhere you like. So I've placed scientific paper so people can see like how do we know these things. Here's what one of the scientific papers talks about the glacier is. You can put photos of the penguins. We've got shown you this yet, CJ, but you're standing here before it's so as he's crossing the Drake Passage, and then you go on a science boat and kind of see the environment, and then you know, you can put up websites, and then you can like say, what's in this ship? Let's head to the ship for another adventure. So the idea, I need to fix this. The idea I've learned that it needs to be more guided, because this is now you enter and you're just swimming off. What do I do? So they've told there's like a rubric, they grade these. So I need to move from the left column all the way to the right column. So I failed this up. But the idea is you would land and you'd have more of like a guided experience. So you're not just going around wondering what you're doing. So then you get on the ship and now you're going to meet and learn about iceberg. You can really put anything. Sometimes the sound. But in those happens look at what we're going to go use this gear bucket. You can see so then you can hop in the boat. You go in the boat. Gear, like it's kind of whatever you want to pick up your participants. So what I want is you to like get inside the boat and then with hot spots. Like, why does this guy have a push penguin? But if you want to go use that, you have to go into the bucket only. Like, you're in this scene, you're going to learn about. Okay, so this is like, what is that doing? What does that data look like? So you get a profile. And then if you want to look at the next thing, you have to the location. Like what this instrument. You want to know what this woman's building, you go locate, and now we're going to do phytoplankton. So what are we learning about the phytoplankton? So I have a lot of fixes to make based on feedback. Kind of skip you. Didn't talk about NASA. I'll fix that. And then you can put fun things like, what the heck's on the horizon? This was an epic moment when an orca was wave knocking seals off, and you can kind of insert fun stuff like that. The scenery's humbling. I don't know. I do have, see, she's doing satellite observations. So I've got a lot to fix on this, but and then Martina's talking I said goodbye, but they asked if I could have a sponsor into the ship. And felt like they were missing the fun about it. So finish that. But then here you could say some experience, we're gonna have links to the website, newsletter, stay in touch, interact with the team. So thank you. Yeah, you can't go anywhere from here. So so ending. But that's what I'm working on. Hoping to have it done before AGU. So. Along with those. But sorry. I got rid of the. Okay. So yes, so the future of Antarctica depends on a generation that understands the direct link to their own backyard. And our projects and collaborations are hoping with the aim to connect people and bridge that gap from the most remote places on Earth to even the tropics. So we're going to complete that digital learning experience, complete the sample processing, of course, put all as the data up in the online repositories, mentoring new students, building new collaborations. Sorry, the floating channel. This Antarctic season starting already, and how to think about how to keep this program going, because yeah, it also has no funding anymore. So the NASA one ended and we don't have further NASA funding. So we're evolving and trying to get creative. We do have a newsletter. There, if you'd like to subscribe, you can. I'll hide this, but there you go. And of course, we try to be active on social media because we don't get to talk to all these people. So we hope to like share as much as we can through that. Thank you. And I will take any questions or discussion or anything. Yes, Sophie. Yeah, can you guys still hear me on Zoom? Okay, yes, thanks, Steve. So yeah, the question was, are they all English speaking, or what are the kind of demographics of language? So mostly it's English speaking. The demographic of travelers in Antarctica you can find on the IATO website. It's mostly US based, with some Europe and South America, Australia and China's growing. So there's a lot of charters, usually that will be Chinese charter specific with translators, but there's also some done in Spanish. German charters, mostly English. There's French companies that go down too. So yeah, I'd say mostly English, but there are other languages. Yeah. Does that answer your question? Oh, yes. So we started off in English, and we are, so if a translator, the companies also sometimes hire dedicated translators, so they'll translate presentations. So sometimes when you're on the ship, you give your presentation, then you have to give it again while the translator's speaking in the ear things. But all of our stuff's in English, but because of the collaborations we've been doing with Puerto Rico and because of our colleagues in South America, we've wanted to expand to Spanish, which we've done now. So we're trying to do everything in Spanish and English. Yeah, we've been asked if we can translate everything into like Russian and Chinese, and we're like, yes, someday. So yeah. Yes, Sarah.
Pay extra to participate in this, or is it included in their package?
Yeah. Yeah, great question. So it's the question was, is it included in the package of their trip, or is this something extra like an add-on they pay to do? From our experience, we offer it at no cost, and all the operators we've worked with, we're aware they also don't offer it at cost. There have been some test things like people don't show up, but you've already like planned the event. So if you charge $5 or $10, like would that incentivize people to be committed to their slot? It's in general, the packages are like all inclusive, and the things you would sign up for to pay extra are like kayaking, snowshoeing, submarine rides. So typically everybody kind of has an opportunity to do the science boat, to do the hikes, to do the Zodiac cruises. But so from what we are aware of, they're not charging for the program. Yeah. Christian.
What are you most excited for about the upcoming field season?
What am I most excited for the upcoming field season? I don't know. I think I'm always excited for the upcoming field season. Like, I really like hearing how people are liking the program, and then they send us photographs of them. So whenever I get a WhatsApp and it's like a spam of photos of everybody smiling, I love that part because it's been quiet, right? Like nobody's been sampling, so my WhatsApp's been quiet. So yeah, I'm excited to hear about your trip down, sending all the selfies. So yeah.
What are like a lot of those bases in the peninsula are like Argentinian or Chilean? Do you see like a lot of like support from like those research institutions, or I don't know, like do they kind of help out a lot, or is it
Yeah.
just we do our own thing
With this program or with science and
with this program?
so actually this is fun. We're starting a new collaboration with the Uruguayan base, Artigas base in Maxwell Bay. So a lot of the ships go to Maxwell Bay because Frey Station and Bellingshausen have not an airport, it's like an air runway, and tourism comes in through there. And so while they're there, Artigas has asked if we could include one of their sampling locations in our efforts. So we'll be collaborating with them and giving that data. So that starts this year. Does that make sense?
Yeah.
Like, and then, yeah, in general, I mean, just in general, working with other researchers, maybe not directly with the research stations doing any of the collection, but more just like as a scientist, we partner and try to collaborate publishing results with other scientists from different countries. So like from British Antarctic Survey, I'm currently talking with about the CTD data and trying to work with somebody who also is familiar with the US Palmer program. Like how could we do some analysis on that? So it's like more in that, the science side of it, not necessarily the physical sampling side of it. But if all the research bases wanted to contribute, that would be amazing, or support in some way, then that could be part of the InSync, like, or the International Polar Year, synchronized sampling from all the bases. Spread the word. Because well, the idea is like, yeah. Oh, InSync. So I'm learning about it still. I guess it's going to be a coordinated effort of synchronized observations and sampling in 2027 to 8, right before the International Polar Year, which is going to be 2032 to 2033. And yeah, so I think it's still in brainstorm phase right now, of what could be contributing to it. And like the idea with FjordPhyto is obviously everybody has their own method of sampling phytoplankton. And with our program, we couldn't rely on liquid nitrogen. We couldn't rely on the samples staying frozen the whole time. And we couldn't rely on anything that took more than 1 hour. So we don't collect things like nutrient data, which need to be frozen until they get back to the lab. We don't collect a lot that maybe in general as a phytoplankton scientist you would want that information, but because it's been tailored to like the least common denominator of sample collection, I think that's what's made it so successful, because it can fit on any kind of application. So the vision would be like if it was in sync and everybody just did like a FjordPhyto protocol collection, then that just continues building that data set. Of course, you could supplement with all the pigments and like everything else that you'd want, nutrient data, stuff like that. Does that answer that question? Yeah.
What happens with the bottles? They're just scooping the surface, floating it. What's the next steps?
Yeah, so the bottle sample is just surface seawater. We ask that they go to their wrist, above their wrist. So they're scooping the seawater, adding a Lugol iodine dye, which preserves the shape of the phytoplankton but kills them as well. And it's non-toxic, so it's not like formaldehyde, which we don't want to be distributing to people. And that sample then just stays, it can be at room temperature or in a refrigerator until they're offloaded in Argentina. And then that gets sent to our colleagues in Argentina. So that's the Lugol microscopy sample. Then the DNA sample, after they filter that material onto the filter, they stick it in the tube with the RNA later. And we ask that we have like a little sandwich Tupperware box. They collect them all in a freezer. So sometimes it can be a ship where the freezer is where the ice cream is, like in the galley. But sometimes they have dedicated science freezers, and it's just a small box. And so all those tubes come back to us. But we ask those stay frozen on the ship until April, when we can offload all that. And then they might be at room temperature as they fly back to the US. But that solution, RNA later, is okay to be at room temperature for, according to the manufacturer, up to 5 days. So, yeah. Does that answer that question? Yeah. Okay.
All right, we should probably wrap up.
Yeah, thank you all. And thank you for the time, attention. Thank you on Zoom. Good to see you all. Oh, hello, Kiefer, Rick, Carlos, Dan, Karen.
We have
Right.
a couple weeks off now, seminar, but then we're back in November with that. So, thank you. Like,
Yeah.
Karen Berg just commented.
Oh, yeah. Karen, can you hear us? What operator is your daughter going with? Yeah.
I think for all of us who are not biologists, the question that we keep asking us, this is asking us, what are the possibilities for net stations or other data?
Yeah.
And
Yeah.
maybe your
That was a big
partnerships.
Yeah.
The pictures.
That would be amazing.
Yeah,
The-
that's the majority of it.
like, It just gives a little more depth into the biological parameter. And chlorophyll was a big, easy one to get,
Yeah.
but like a little more depth in that. A phytoplankton.
You know,
So,
as soon as,
yeah, how do
I
I?
get back, I definitely talked to you about it like last year. I
Let's
wanna
just
say
also sharing
Yeah.
So.
Yeah, yeah,
share the data.
yeah. I think,
I can just go to the biology.
So.
yeah, there's definitely a lot of interest from them. There aren't enough projects for them to keep people busy. And so they are always looking for stuff. Yeah, but
On the page.
yeah, but it would be
But there's also the
helpful.
question, people, they do something that's just because they have to do it.
Yeah, yeah, so that's where there's not a good system yet for like integrating, because right, like, let's say you could get to a website and have almost like a shopping cart option of programs you want to run, or you're like, I want to do the phytoplankton sampling, or I want to do today we go out and do the anemometer sampling. Yeah,
And you gotta have an anemometer all the time, but you can talk about it.
yeah, so getting the logistics figured out, but that should. Projects. Cause like they can only, especially if it's bad weather, you can't take the boat
Antarctic
down.
survey people.
Yeah.
Yeah.
Or maybe it could even go through IATO as like a broadcast, or CC even, because I know they try to tap into those. Yeah, we can be in touch and, yeah, okay, sure. Thank you. Hello, Mary. Good to see you.
I always try to read through the